Good afternoon. And yeah, many thanks for getting me here to Jürgen, Christoph, Juan, and particular thanks for the very kind words. Bottom line of what you said is essentially it pays off to be kind. Memorize that. I feel in this audience I'm a bit of an exotic guy. Christoph already mentioned that I'm doing molecular dynamics simulation as our working course. And I guess there will be a couple of students who don't know exactly what this is all about. And so I thought I'd give a short introduction in how this method actually works. The main part will be, depending on time, one or two examples of how we apply it. And then I will give a sneak preview in this very nice collaboration, which I enjoy a lot, which we have here with the guys here. All right, maybe one sentence to memorize would be all the dots which you see on your nice graphs in bioinformatics and proteomics. Each of those dots is actually a molecular machine. And we are interested in particular examples of these molecular machines, how they perform their particular task, what's the trick nature has up its sleeve to actually perform a certain function, typically much more efficiently, better than we would be able to come up with. So that's our main goal, to understand them and to understand these machines from a physics perspective, so to say, from first principles. To give you an example, what I mean is by these molecular machines, that is one of my favorites, F1 ADPase or F ADPase from mitochondria. It generates all the ATP in your body, many of you will know that, about your own body weight per day, right? You don't gain weight, don't be afraid because of that, you use it up again. But that's about the consumption of ATP of your body. And without going into details here, just as an illustrative example, this thing works very similarly to an auto engine which you have in your car, a combustion engine, right? So we have a crankshaft here, like this thing here, this orange part here. We have pistons moving out and in. These are these parts here on the right side. And the only difference is it's fully reversible and it has an efficiency of nearly 100%, whereas our macroscopic machines double around at maybe 20–25%. So there's a lot of motion going on when you achieve this function. Another example I'd like to share with you, and I will get back to that one, it's the ribosome, many of you know that. And just from the sequence of cryo-EM structures, that's not the simulation yet, but you can probably appreciate how dynamic this whole machine is. It's a huge molecule and it's super dynamic. We want to understand this dynamics. And of course, it's clear that these machines don't work in isolation. I think I don't need to tell this audience. It's a whole production line of machines which firmly cooperate very closely. Okay, having mentioned that, how do we approach it? So what we try to do is to calculate the motion of every single atom of what we call a simulation system, which typically consists of a few molecule proteins, in this case aquaporin. The water channel sits in a membrane, so we include also membrane, a patch of a membrane here in our simulation system, atom by atom, as well as water. So this sprinkle here, which you can see here in reddish, whitish, so that is water. If you want to study water conduction, you better also include some water in your simulation system, right? So typical size of such a simulation system, like a couple 100,000, today up to several 10 million atoms, which I guess longs for a large computer. How do we proceed? Well, it's very, very simple, in fact. So we calculate at a certain time, and we know all the atomic positions, or our computer knows those. We calculate the force, if you zoom in here, the force onto every single atom. And this force arises from long-range electrostatics interactions, Pauli repulsion, that's the force which prevents us from just falling through the ground. And all the chemical binding forces, chemical bond lengths don't want to extend too much, angles don't want to deviate too much from their equilibrium angle, and so on and so forth. So we know all these molecular interactions, interatomic interactions pretty well, and we can program our computer so that it calculates it for us. Once we have these forces, we go back to Sir Isaac Newton and ask him what to do with those, and he will tell us, aha, they accelerate our masses here. And so the atoms get into motion, and after a very short time, they will have moved a little bit, and therefore the forces will also have changed. So guess what? We need to ask our computer again to calculate the forces again. And you can see where that leads to. We progress in time in very short, as we call it, integration time steps, typically 10 to the minus 15 seconds, pretty short. So we have to carry out them over and over and over and over and over and over and over again to eventually end up with what we call a molecular movie, right? So that would be the raw data of such a simulation. You can see the self-diffusion of the water. If you would be patient enough, you would see water going through there. Actually, that is one part where we got into contact with experiment. By just counting the water molecules per microsecond or so, we can calculate the conductivity, and that can be measured. And we got it right to about 20% accuracy, which for a rate is actually great accuracy. This study also predicted a number of mutations, and then predicted also how the water permeability should change. And that also was then confirmed experimentally subsequently. So this is how we interact with experiment. We try to calculate something which can be measured. And then if it agrees, hopefully, then we can calculate something which people cannot look into, in particular, how this water conduction really takes place and how nature makes it, that this protein makes it so efficient and selective. It's just one example. Another example, which goes actually also back to Munich here, in collaboration with Helmut Grubmüller, a long time ago, he had the idea, why don't we try to simulate atomic force microscopy experiments he did in this lab? He was able to, at that time, spectacularly rupture single molecules. In this case, it would be a biotin molecule ruptured out of a binding pocket to which it is non-covalently bound. In this case of streptavidin, that's a very well-known biotechnological protein. And he was able to rupture this, to drag the biotin out of the binding pocket. And we did the same in the simulation. So we applied some mechanical force to this particular atom, which is the same as in the experiment. And then you can probably see, you may see in textbooks, this binding, unbinding energy landscape. It's a bound state, a well, a barrier, and then the unbound state. You can see from here, it's way more complex. First of all, you can see a very rugged energy landscape with many local minima and maxima. And second, if you repeat that, you can see that the biotin flicks out of the binding pocket not in one rush, but in many intermediate steps, which are different from attempt to attempt. So you get stochastics in there. And at the end of the day, you have a maximum rupture force, which you can compare. I'm showing you that also to show very briefly how the field has developed. So this is roughly a little less than 30 years ago. And what we did there is we pulled the ligand out at different velocities. You can imagine, the faster you pull, the harder you pull, the more force you need to get it out. Actually, if you let it just sit for a couple of months, it will get out spontaneously by activated processes. And our friends from the AFM did exactly the same thing. They also pulled a little faster and slower. Notice the logarithmic scale here. And yeah, you can see it, how that depends on the velocity. That's the experiment. That's our simulations. We couldn't get faster at that time simply because our computers were not fast enough. Yeah, we could simulate something like 10 nanoseconds or so. And our friends haven't got up. They couldn't get, we couldn't get slower. They couldn't get faster because the whole thing is limited by the resonance frequency of the cantilever. So that's the best they could do. That's the slowest we could do. And you may see there is a huge gap at that time. And to say that the two agree is a long stretch, I would say. Nevertheless, good old times was enough to publish it in Science. Today, we revisited that 2 years ago or so. And now our friend Simon Scheuring was able to really go up the way. Here in blue is the experiment again, up to here, whereas with new computers, generously paid by the Max Planck Society, we got down to here, and you see a nice overlap. And now I think it's convincing to say that experiment and simulation really agree nicely with each other. And I should really emphasize there's not a single fit parameter here, right? Either it fits, it matches, or it doesn't. And in this case, it matches wonderfully. I still find this amazing for such a complex system of several 100,000 interacting particles that at the end of the day, you get a number and it agrees with experiment. The physicist amongst you will also say, oh, well, but the real thing is to predict something. Have you also been able to predict something? Yes, we have. We saw in our simulation these intermediate states briefly before rupture. So that's an exit state, into metastable state. And we saw it clearly here in the simulations. And then we asked our friends, please have another look in your data. And lo and behold, they also saw it in the data, albeit with certain lower time resolution. So yeah, we can predict things. So with that, I hope you get in a nutshell idea how actually our simulations really work. And I'd like to talk about 3 projects now, see how time progresses. The first, I mentioned ribosomes, and we will have a particular look at how the peptides are synthesized and how the nascent peptides go through the nascent exit tunnel and how antibiotics work in this exit tunnel. And then if time permits, I want to share little new results on microtubules. And I will definitely want to share first results from our collaboration with Christoph and Jürgen and Juan here on a simulation of a whole, if you want, a drift chamber of which you know much more about than I do. You can see a pattern here, right? So we try to really simulate experiments as closely as possible, one-to-one, to really see first that we are correct, and second, and accurate enough, and second, to provide additional interpretation of the experiment. So let's jump into it. For those who haven't seen ribosomes yet, I just want you to know that here we have transfer RNAs which bind to an A state in the ribosome. Then they move over to the P state, and there they deliver a peptide and an amino acid, which is attached right here to the growing polypeptide, which eventually, when it exits the tunnel, becomes a new protein. And then the tRNA leaves, and it binds so with a triple nucleotide triplet, yeah, to the mRNA, which encodes the particular protein. So for each triple, you have a specific tRNA, which just carries the right amino acid here fitting to the genetic code. And that's actually how the genetic code is really implemented. We were quite curious on what actually happens in the exit tunnel. And for 2 reasons. First of all, from a physics perspective, it's quite amazing. Have you ever tried to kind of push a cooked spaghetti through a winding road tunnel or something, right? So it's quite hard. To pull, it's easy, but to push, that's actually what happens here. And we were quite interested, and this is an ongoing project, how this actually works. And second, many antibiotics work right exactly in this tunnel. They bind somewhere in this tunnel, and we would like to know how they work. You probably also know better than me that most drugs and antibiotics, we don't know yet how they actually work. And obviously, it would help to improve them to know how they work. That is a short simulation which just illustrates how we actually simulate the adding of one after the other amino acid at what is called the peptide synthesis center, PTC, or peptide transfer center. And this is the growing nascent chain. In this case, with a dye tip, because again, we collaborate with people who can do spectroscopy here, and you can nicely see how this grows through the nascent chain. And now we actually teamed up also with a group here in Martinsried, Dan Wilson, who's now in Hamburg, and he was able to do a cryo-electron microscopy picture of a bound antibiotics here in the exit tunnel. So here again, this is a tRNA, and now the exit tunnel runs towards the bottom here, and the red thing, this is now erythromycin and antibiotics. And the common notion more or less was just that, well, it's pretty easy, right? So this thing binds in the tunnel like a cork in a bottle and blocks the tunnel. And so the nascent peptide can't go out. That's how it works. Well, it was found early on that not every peptide is actually blocked. So some sequences can actually go through and others cannot. So the whole thing is sequence specific. And of course, the question then was, well, that cannot be just a simple cork in a bottle. How does it work then? And gradually people came up with sequence motifs which were able to pass and those which stalled. And with that structure, we repeated the whole thing in the simulation. So here's the bound erythromycin. And we removed it in the simulation and saw and checked what happens also as a function of the sequence of the nascent chain. And long story short, if we now zoom in here in this part here, where the amino acid here, which still is attached to the tRNA, actually gets synthesized with a nucleophilic attack to the growing chain here. When you look very closely at it, then what you see is that as soon as this thing here, for example, if this is a lysine with a positive charge, it is kind of dragged to this position. And this red here is the structure with the erythromycin, and the green here is the structure without the erythromycin. So what happens is actually that this erythromycin induces an allosteric structural transition all the way up here, 5 nanometers up here, which changes the backbone here of the ribosomal RNA. And with that, also changes the stereochemistry, in particular the distance between the attacking amino acid and the recipient of the amino acid here at this site. So it perturbs the whole stereochemistry. And this is such as what we proposed from the simulation. This is why actually this antibiotics stops or stalls translation, and why it is also sequence specific, because if there's no positive charge here, that doesn't happen. So it needs a positive charge. Okay, control. What if we have no charge? Okay, nothing happens, fine. And that, of course, brings up the idea, let's put another positive charge here, arginine, for example. And strangely enough, this hasn't been tried before. So let's put that here. And yes, again, we see the same effect. Not going into detail here. The important thing is, then our colleagues in Martin's Reed did the experiment, and lo and behold, yes, the prediction again came out to be true. Also, arginine blocks, stalls, if it's at this position, the peptide synthesis, and even more so even than lysine. So we have a new mechanism how this particular and some other antibiotics actually work. By allostery, not by blocking the exit tunnel, but by perturbing the stereochemistry of the actual peptide synthesis step. And since then, we have looked at a number of antibiotics, and it seems to be a common theme that actually wherever it binds, at the end of the day, it acts back on the PTC, on the active site, and perturbs the stereochemistry. So this is just a few examples we have now looked more recently. Okay, so that was one idea or one topic question which we were able to address with simulations. As a small detour, let me maybe briefly look at that picture together with you. You know those pictures pretty well, I guess. These are single molecule cryo-EM images from which you can not only extract a structure, but also an ensemble of structures. So the idea is at room temperature, proteins move quite a bit, as I've shown you. If you shock freeze them fast enough, then you preserve this distribution of conformations, and you can look at them and sort them out in cryo-EM. Question is, of course, when they shock freeze or plunge it into liquid ethane, like in that preparation step. So here you got a grid, and you put solvated, for example, in this case, ribosomes, the red dots here on the grid. They end up in these small holes embedded in a very tiny sheet of water, and then you plunge it into liquid ethane and it becomes ice. And the question is just how much of the conformational dynamics at room temperature, at ambient temperature, is actually preserved when you go down to 90 Kelvin or something. An indication that it is quite a bit is actually if you compare the B factor, so that is the how much motion is actually a structural heterogeneity is seen in the cryo-EM. That's the colors here. And when you compare this with the motions you see in the molecular dynamics simulations, the amplitudes of those motions, then you see a certain correlation, albeit not perfect, right? So there's also regions which seem to move in the MD, but not so much preserved in the cryo-EM. So again, to what extent does such a cryo-EM ensemble actually reflect the ambient temperature conformational dynamics at in reality at room temperature? And to that, just a very short intuitive picture here. So you may know that proteins can adopt many different conformations. And the reason is that the energy landscape of protein motions, which is a super high dimensional free energy landscape, has many almost isoenergetic minima. And depending on in which minima the protein actually ends up with, it adopts a different conformation. At room temperature, all those almost isoenergetic minima are more or less equally populated because the temperature is high. And if you would now shock freeze instantaneously to cryogenic temperatures, you would preserve this, of course. But instantaneous is not possible. So what if you shock freeze a little slower? Well, then there will be enough time for those guys here, at least some of those guys, to overcome those barriers, and they will go towards the lowest minimum or to the lower minima. And so the whole ensemble will kind of shrink towards the lower energy regions of conformational space of this energy landscape. How much? That was the question. And to that end, we first estimated how fast actually will this plunge freezing be. And it's hard to really find out, cannot be measured, but we came up with something like a couple of microseconds. And that is nice because this is exactly what we can also do in the simulations. And therefore, we just set out to do this shock freezing on the computer. And you can see here what actually happens. So here's a zoom in of this part here. And you can see at room temperature, it's highly mobile, but then it kind of arrests into a quite rigid structure after shock freezing. The whole thing is in water, so we just don't show the water. It's always water. And now, after quite a lengthy analysis, which I will skip here, our main result of that small detour is that about 20 to 30% of the fluctuations are lost in the process. But the good news is, with our simulations, we can now recalibrate the cryo-EM data and recalculate back to room temperature and calibrate or renormalize the ensemble such that it better reflects a room temperature ensemble. And for the experts, so in the process of shock freezing, barriers of about maximum of 10 kilojoules are actually overcome, and higher barriers are already trapped, so it's fast enough. So that was Lars Bock's work, who's the specialist for ribosomes in my group. And Maxim Migayev, he has looked at microtubules and microtubule stability and mechanical properties. And you may know that microtubules are the railroads of the cell. They form long tubes, and they can assemble and disassemble. And typically they grow, and in growing, they also can exert forces. So it's kind of a linear motor. They can also, when they disassemble, that happens typically pretty fast. So it's called catastrophic decay. And this disassembly actually can also act as a molecular motor, like indicated here. So if you put a ring on this end of the microtubule and you can see it disassembles, that will push the ring upwards. And this is exactly the linear motor which separates the chromosomes during fertilization. So if during mitosis, when the chromosomes have to be pulled apart to be separated for replication. And this is exactly the machine which does that. And the question was, what switches, how does the switch between growing and shrinking actually work? How is this controlled on a molecular level? And there, the previous picture, the initial picture was pretty simple. If the kind of the end of it's kind of a chimney, right? The end of this tube, imagine a chimney with bricks. The bricks are the tubulins, the individual proteins. If this is intact, then the individual proteins can assemble quite easily, like here on the left side. And things work out and things grow. Whereas if the whole thing is splayed like this here and the microfilaments splay outside, then it will decay because then the tip is unstable. The whole thing is, by the way, regulated by GTP hydrolysis. So this would be more GTP-like, and this would be after hydrolysis. So that was the view held until recently, when certain McIntosh and others were able to do very high quality cryo-EM images of growing and of shrinking microtubules. And I can stare at this as long as I want. I at least don't see much of a difference here. So there is also splaying here, the same amount as here. I don't see much. So this cannot be the reason. And this is what we looked into by simulations. And we looked into how the monomers actually undergo a kinking motion upon hydrolysis of GTP. We looked at the lateral interaction between the motions, and then we put everything together into a simulation of the whole microtubule tip. And this is what I would like to show you here. Just focus on the third part here, and just sharing with you one of the largest simulations which we carried out, so 15 million atoms, and there you can see very nicely displaying of this tip. So it doesn't stay intact, and we did that with GTP loaded and with GDP loaded microtubules, and then looked at the differences. And again, you can probably already see here is displaying much more pronounced than here. It also will progress, but much slower. And at the end of the day, again, just giving you the bottom line for time restrictions, what comes out of this simulation is a tug of war between the bending outwards of the microfilaments, like that. And you can see if you are an intact tip and you want to bend outwards, you have to split the lateral interactions, right? So the contacts need to be split. And this is exactly the opposing force to the bending. So we have the lateral interactions and the bending being in a tug of war. And when GDP hydrolysis shifts this bending back and forth, and therefore affects the growing and shrinking. Now, one particular prediction of this simulation, you may have seen that already in the movies here, is that there will be a number of microfilaments always remain in contact, like those 4 here, or those 2 here, and the others will split. And those physicists amongst you have been studied ferromagnetism. It's the same frustrated energy landscape idea which leads to the formation of these so-called Weiss regions, which you find in iron, in the ferromagnet, right? So they try to minimize this tug of war energy frustration. Same thing here. So it's interesting physics. But the question is, of course, is that correct? And again, very recently, we were quite happy to see another in the Dr. Room lab, another cryo-EM study where you see nicely these aligned microfilaments precisely as our simulations were predicting them. All right. So the last 5 or 10 minutes, I'd like to spend on work by Daniel Schölechi, and that's the work I mentioned in close collaboration with Jürgen Cox-Juan and Christoph. And the idea was simply, at least one of the ideas was, can we really simulate a drift tube in a mass spectrometer, right? Can we simulate that? You all know that there is a very simple way to calculate the mobility, the ion mobility, just from the cross sections. So if you have the structure of the peptide, then you do the cross section, and that gives you a rough estimate on the mobility. This is how it's, I guess, routinely done. And I really don't need to tell you how a drift chamber works. You know that much better. This is, by the way, a simplified version. I learned now the hard way that it's actually more complicated to get higher accuracy. But this is our Mickey Mouse picture, which we simulated. And of course, there's an electric field accelerating the charged peptides through very diluted air at a couple of millibar, and the air resistance, of course, works against that. So I'm carrying O's to Athens, I guess, here. But can we simulate this thing? And one thing which Jürgen came up with was that, strangely enough, and I'm only showing here a very schematic view, there are, for many peptides, 2 peaks, two distributions, 2 modes of mobility, a high mobility and a low mobility group set for one and the same peptide. And this is seen for many different peptides, so that seems to be a common thing. And the question also was, why is it that we have this bimodal distribution? Why don't peptides show a whole variety of conformation? Is it actually conformation related, or does it have some other reason? And so this is one of the questions we addressed. The other would be, now from me being a physicist, I would be very curious, I was super curious, how does the interaction with the gas actually work on a molecular level? After all, the gas molecules impact our peptide with super high velocity. Does that harm the peptide? Does it change the structure? Does it promote conformational changes? How does the collision work anyway? Is it an elastic collision, et cetera, et cetera? And also, can we improve this estimate of the, I would call it a CT value, right? The ion mobility coefficient. Can we improve on that? And to that, I'm we did two sets of simulation. The first is we took a number of the peptides which Jürgen and colleagues had in the drift chamber and simulated them in vacuum. So let them fold in vacuum. And here you can see one of those simulations. That's the peptide here. This is for the experts, how we carried out the simulations. So you can really see that curl up very quickly into an alpha helix. And that's one thing which can happen. And if you do that many, many times, we did that like 1,000 times for this peptide. And over time, we did here the radius, the histogram of the radius of gyration. Then we immediately saw that we have, in fact, a bimodal distribution at the end, right? So we have a large radius of gyration species, that's actually this one, and a small radius of gyration species, which is actually a more globular, curl up, fold, molten globule type thing of the protein. So they adopt quite different structures from in solution, but this is what we see. And we see that over and over again for many different peptides. And again, here quantified here in this histogram. So that looks promising. But now the question is, of course, does that match the bimodal distribution which you guys saw in the drift chamber? And to that end, we actually embarked on simulating a whole patch of this gas. You can here see the gas molecules, nitrogen and oxygen, a little bit of water in it. And here's our peptide. And actually, you don't see much here, right? So because a gas is so diluted that it rarely hits the peptide. And actually, this is also super slow motion, right? So what you see in every second is just 30 picoseconds here. So not much happened. Let's speed up a little bit. And then you see occasionally a gas molecule actually hitting our peptide here and getting it into motion. And also the electric fields starts to act. One thing which surprised me is quite a bit, it doesn't just accelerate it in one direction in a more uniform motion. It's kicked around quite a bit on the microscopic scale. And of course, that reminds you of Brownian motion, right? So this is exactly what happens here. And it doesn't always move in the quite right direction, but on average, you see a superposition of this direction in the right speed. I'll get back to that in a minute. But first, I'd like you to just marvel at the collisions which we see. There are several types of collisions, like this one. I'll do it again. So that is what I would call an elastic collision, a gas molecule. You can see how fast it actually is according to the Maxwell distribution. Hits the molecule and just gets bounced. So it really gets elastically nicely bounced off. Another would be what we call a kind of a swing by. So it goes from the other side and actually gets attracted. And that's quite remarkable because it's electrostatically neutral. It's just an oxygen atom. But the van der Waals interactions are strong enough at room temperature, that's a surprise, to actually bend this trajectory and transfer some momentum also to the molecule. And the last one surprised us most. That's kind of a absorption and re-emission, right? So the molecule gets here and it actually binds to the surface of our peptide here. And only after a while, here it is, and then it gets re-ejected in a different direction. Quite amazing. And so we have three different elementary processes which actually impact some velocity on the peptides or slow it down, depending on how you look at it. I was mentioning this is quite a stochastic process, and what you can see here now is different simulations, 10 different simulations in different colors. And you can appreciate if you wait long enough, we didn't wait so long here in the movies, but if you wait long enough, then your peptide gets accelerated. So this is the velocity here in meters per second, quite a bit, until it reaches a kind of a terminal velocity, which is exactly what you observe in the drift chamber and what you also know from parachuting. So at some point, the air resistance equals the force, in this case, electrostatic field, or for parachuters, the gravity, of course. And if you fit the respective dependency here on these bunch of curves, the red is the average, then you get a very nice number for the terminal velocity. And just to show you, we did that over and over again, also for different peptides and also particularly for different conformations, because at the end of the day, we want to see how much these conformations affect the drift velocity. And long story, well, I shouldn't say short, it's an ongoing project, but where we are now more or less is that we actually get helical, that's those two here, and globular, the other ones, structures here. We get different drift velocities,
And this is what we get from molecular dynamics here, these numbers in meters per second, quite distinct here. And this is what in the experiment is actually measured. And I should say, I think, correct me if I'm wrong, but I think it's fair to say that the main reason for this difference is actually that we don't know precisely the air pressure in the drift chamber, the density, and also the electric field is not so easy to grasp because it's a non-homogeneous field in reality in a real machine. And so I would think this is the main reason for the difference. If this is the case, then the ratio of the two should agree much better and actually does within error bounds. So this is where we are now. I think it's fair to say that we now know a little better how these drift chambers actually work, the collisions with the peptides actually look like. One result was also, apparently, despite the high impact velocity, the structure is not much perturbed during the collisions. So that is also one reason why you see two different distributions. They don't interchange in the drift chamber. Either you are stuck in the helical conformation as a peptide, or you are stuck in this globular conformation. That's what we also see in the simulations. 2 minutes of perspectives. Where are we going? Is that fine? Yeah. One direction, and that is closely related to what we see here, is we are particularly interested in proteins which do not adapt a native structure. And here in vacuum, they already don't adapt a native structure, but there are also proteins who don't do that in ambient and solution conditions, so-called intrinsically disordered proteins. That is one direction of research where we think that molecular dynamics can help particularly well because they are particularly hard to measure. Nobody knows really how they undergo their structural changes. And here you can see such a thing on the right and on the left side. And you can probably see the huge difference between those. About 30% of your proteins, mostly cancer related, are actually those guys here. Another one direction which we're going is towards pharmacology. You all know that in industry, drug docking approaches are heavily overpopulated, I should say, and not super accurate. We think that molecular dynamic simulation can do better here, albeit at a high computational cost. And you can see here, for example, the binding of a drug candidate to the binding pocket of a particular protein. And you can see when it binds, these two lobes kind of close like a fist over the two. And this induced fit motion is precisely what is not captured by traditional approaches. And we think we can help with molecular dynamics. Also, replacing all these huge robot lines of assays probing for drugs, at least in part. Finally, we are quite happy that we are able to go to larger and larger systems. The largest system we had so far is a whole virus capsid, and we were looking at how the virus capsid changes its structure upon calcium addition. You know that then afterwards the genome, the viral genome is released, the RNA is released. So that was one of the projects. Eventually, we can go to whole organelles. And if for the philosophers amongst you, I think it's quite amazing because I didn't expand on that, but all what I was telling you, all those simulations rest on one single equation, and that's the time-dependent Schrödinger equation. We do a number of approximations. I didn't have the time to expand, but at the end of the day, it goes back to this equation, and that gives rise, both historically, but also in length scales, to the behavior of water, the properties of water molecules. First, proteins being simulated, these nice machines I was sharing with you. Membrane fusion, viruses, microtubules, maybe organelles. And I don't know where we're going to with faster and faster machines and better algorithms. I think it's fair to say we're living in exciting times, and we get a foothold on understanding life processes from basic fundamental physics principles. And that is something which I find highly exciting, and I'm particularly thankful that I'm able to take part in this endeavor. I'm not reading down the summary. I'm just letting it here to let more time for questions. I just want to thank those guys and my group, without which nothing of that what I was sharing with you would have happened. And thank you very much for your kind attention.